All right, so it's my pleasure to introduce our second speaker of today, uh, Courtney Harrison. She received a BS in Animal Nutritional Sciences from West Virginia University in 2018. And after graduation, she worked at the Conservation Fund Freshwater Institute in Shepherdstown, West Virginia as a research intern. She then went to Michigan State University in the spring of 2019 and is now a third-year master's student studying the role of infectious diseases on the health and recruitment of juvenile Great Lakes lake whitefish. Take it away, Courtney.
Thank you, Nick. Can you hear me okay?
Yep, sure can. The screen looks great.
All right, perfect. Thanks. Thanks again, and thank you to the organizers for having us all come together again and present virtually this year. I also want to thank the co-authors of this study and Dr. Brendan and Mark Ebner from the Quantitative Fisheries Center here at MSU, and Isaac Standish from the La Crosse Fish Health Center. I'd also like to thank all of my colleagues at the MSU Aquatic Animal Health Lab. So today I'm really excited to present some research findings about a particular bacterium to be a possible factor in poor Great Lakes lake whitefish recruitment. So for those of you that are less familiar, this is the lake whitefish, Coregonus clupeiformis. It's one of the few truly indigenous salmonid species here in the Great Lakes, and it's believed to have been around since the glaciers carved out the Great Lakes. And you can see by its downward pointing mouth that it's a benziferous fish species, so it plays an important role in cycling energy from the lower trophic levels to the higher trophic levels in the Great Lakes. It's also the most commercially important fish species in the 4 lower Great Lakes. And so here you're seeing lake whitefish commercial yields over time, and you can see that their numbers have fluctuated. So they reached an all-time low in the 50s, largely due to invasive species and habitat degradation, but with better management practices, their numbers recovered. However, since the early 2000s, their numbers started declining again. But these trends are not only limited to adults. These trends are extending to early life stages. And so these early life stages of lake whitefish are suffering from poor recruitment. And so that's what I'm trying to tackle here. So before I move on, I just want to make sure we're all on the same page about what recruitment means. So simply put, it's the introduction of new individuals to a population. So that means that eggs need to be successfully laid and fertilized, they need to hatch, and then the fry need to survive in order to join the adult population. So seeing declines in the recruitment of young lake whitefish is concerning, especially because we don't know what's causing these declines. And so based on many years of data and studies, there's a number of things that could be happening, such as abiotic factors like climate change and ice cover. And then there's some biotic factors in play. For example, invasive species. And then there was a decline in the primary prey species of lake whitefish. So although these studies were significant, there's still no conclusion as to what's driving these recruitment declines. And so in a nutshell, we have these abiotic and biotic factors interacting like this, so like a stressed host and then suboptimal environmental conditions. And so this presents the perfect opportunity for pathogens to do their most damage and cause disease. And so disease as a factor in poor recruitment has not been thoroughly investigated. And so several pathogens have been found to infect adult Great Lakes lake whitefish. So these papers that you're seeing here are nearly everything what we know about diseases affecting Great Lakes lake whitefish. However, they're only focused on adults. And to this point, we found the pathogen in these fish, and we've also found signs of disease in these fish, but whether the pathogen is causing disease in these fish is still unknown. And so another complete unknown is if these pathogens are present in the reproductive tissues of spawning fish. And if they are present in the reproductive tissues, does that lead to infection in the eggs? And if the eggs are infected, does that persist to infection in the sac fry? And likewise, would those infections persist in later early life stages? But above all else, what are the effects infectious diseases have on the health and survivability of the host, the lake whitefish? And so to put all of this into context, and if you recall from my presentation that I gave last year, I presented my master's thesis concept. So I'm casting a wide net looking for a multitude of pathogens in spawning phase adult Great Lakes lake whitefish. And so in order to do that, I focus on analyzing spawning phase adults. So to understand this, and to understand poor recruitment, I worked closely with Mark Ebner, and based on decades of data, I selected paired sites in Lake Michigan and Lake Huron. So in doing that, one site is associated with good recruitment. So it has historically high biomass and abundance. And then the other site is associated with poor recruitment. So overall less biomass and abundance, and also evidence of fewer early life stages of lake whitefish. And so in collaboration with charter boat captains, I collected 60 fish from each site twice over a period of 2 years. And so I recovered a number of pathogens that we're all very familiar with. And that's where I left things off last year. And so due to a series of events I'm about to tell you, I want to focus the remainder of my time today on this bacterium in particular. So just a brief overview of Corynebacterium and other salmonid species. It's a gram-positive bacterium within the family Corynebacteriaceae. And for those of you that attended the Fish Health Section business meeting a couple weeks ago, you recall Dr. Ken Phillips talking about how Corynebacterium is an emerging issue in the Midwest, especially in hatchery systems. And that's because this bacterium causes pseudokidney disease. It causes mortality, including post-spawning mortality in other salmonids. And it has a penchant for reproductive tissue, so it presents the possibility of infected parents infecting their offspring. And so finding Corynebacterium in lake whitefish is especially concerning because juvenile recruitment is so poor. And so with that, I formed an overarching hypothesis that Corynebacterium infections are negatively affecting Great Lakes lake whitefish recruitment. And so to address this hypothesis, I have these set of objectives. So one, to elucidate the prevalence of Corynebacterium infections in wild adult lake whitefish, including the reproductive tissues, elucidate prevalence of Corynebacterium infections in wild juvenile lake whitefish, and then determine the pathogenicity of Corynebacterium in juvenile lake whitefish under controlled laboratory settings. And so for now, I'm going to focus on this first objective. So during the course of clinical exams and analysis on 480 fish collected from both good and poor recruitment sites in Lake Michigan and Lake Huron, I noted some gross signs of disease, like this one you're seeing here. This fish has some multifocal intraocular hemorrhaging. And then I also noted some internal signs of disease. So a subset of adults had severe hyperemia of the swim bladder vasculature. And so whenever I opened up these swim bladders, I saw this mucoid turbid fluid. And then in some instances, this fluid was more severe than in others. So this one here, the fluid is filling up almost half the space of the swim bladder lumen. And now you're seeing a primary culture. So I actually took 10 microliters of that swim bladder fluid and plated it onto sheep's blood agar, and it yielded this intense pure growth of one colony morphology. And so remember, the study is focused on recruitment, so I definitely wanted to look at the reproductive tissues of these fish. So I looked at the testes and ovaries of these spawning adults, and I took bacterial cultures of each. And surprisingly, I found the same bacterium in the reproductive tissues. So not as intense as the infections that I recovered from the swim bladder fluid, but still the same morphology from the testes and ovaries of these fish. So now the major goal moving forward is, what is the identity of this bacterium? So I verified culture purity, and I performed phenotypic and biochemical characterization on several isolates. And in all cases, these isolates were gram-positive, oxidase negative, and catalase negative. And I also ran a battery of other tests, but I won't go through all of those with you today. But I did come to a presumptive identification of this bacterium belonging to the genus Corynebacterium. So then to confirm its identity, I ran PCR on the 16S rRNA gene, and then I purified the amplicon and performed sequence analysis. And so this analysis placed the bacterium I recovered into the genus Corynebacterium. So now I wanted to look at phylogenetic analysis. So what you're seeing here is a dendrogram of all described type strain Corynebacterium species, including mine. I color-coded to depict the year the isolates were recovered. So yellow coming from 2018 and green came from 2019. And then the shades depict the location that these were recovered from. So you can see that I recovered isolates from both Lake Michigan and Lake Huron. But the big takeaway of this is that compared to all described Corynebacterium type strain species, all of my isolates fall into a robustly supported clade that contains the type strain of Corynebacterium. So that means that all of my isolates are Corynebacterium multirhematicum. But how prevalent were these infections? So now I'm showing you Corynebacterium infection prevalence in adult Great Lakes lake whitefish from 2018 through 2019. And I'm also showing good versus poor recruitment sites within each lake. So first, let's look at Lake Michigan. So in 2018, I only found evidence of Corynebacterium infection in the poor recruitment site. And in 2019, I found the same thing. So this held constant over 2 years. And in Lake Huron in 2018, I found a prevalence of almost 7% in the poor recruitment site. And then the prevalence in the good recruitment site was half of that. And in 2019, the prevalence was equivalent between the two sites. But a big takeaway is that 3 out of the 4 paired comparisons, the bacterium was more prevalent in the poor recruitment sites than in the good recruitment sites. Okay, so to summarize the findings from the adult fish, I found Corynebacterium in fish showing signs of systemic disease. And for the first time, the bacterium was found in the reproductive tissues of male and female spawning fish. And Corynebacterium infections were either not detected in good recruitment sites or they were found at a higher prevalence in poor recruitment sites. And so now that I have a better understanding of Corynebacterium infections in the adults, now it's time to look at the juveniles. So I had plans to go out into the field and collect wild juveniles via beach seining in 2020. Unfortunately, that didn't happen due to the pandemic. So I was hopeful to have some sort of field season this year in 2021. And so working with folks at US Fish and Wildlife Service in Green Bay, we were able to collect over 100 wild juvenile lake whitefish. And luckily this site corresponds to the good recruitment site in Menominee in Lake Michigan. And so I recently did this sampling and I'm still working on identifying bacteria collected. However, based on morphological and biochemical characterization testing that I've already completed, there are no bacterial isolates consistent with the genus Corynebacterium. And even though I was only able to sample one site into this year, yet again, and like we saw in the adults, I did not find Corynebacterium in the good recruitment site in Lake Michigan. So now the question is, what capacity does Corynebacterium have to negatively affect the health of juvenile lake whitefish? So now I set out to conduct experimental challenges under controlled laboratory conditions. So in order to do this, I needed juvenile lake whitefish, and I was unaware of any captive lake whitefish broodstock that could serve as a source for gametes. And because of that, I collaborated with the Little Traverse Bay bands of Odawa Indians, and I received juvenile lake whitefish. So these eggs were artificially fertilized from wild broodstock originating from Elk Rapids in Lake Michigan. The eggs were disinfected with iodophore and then water hardened with erythromycin on site, and then once again disinfected with iodophore before entering their hatchery facility. And then once they hatched, they stayed at the hatchery for about 3 months, and then they were transferred to our lab and held under quarantine conditions in UV treated water. And so I continued to maintain these fish. Unfortunately, 2020 reared its ugly head again, and I was about to initiate challenges. So in the process of, the pandemic, I continued to maintain these fish and they continued to grow. And unfortunately, in October, I started to see the development of disease signs in these fish. So I started seeing some linear ocular hemorrhaging, severe exophthalmia, and then I started seeing even more severe ocular hemorrhaging. And as things progressed, I started noticing more lenticular and corneal opacity. And then here I'm seeing lenticular opacity, but now with corneal perforation. And when I opened up some of these fish, I saw some visceral edema as well. So collectively, this indicated that these fish were going through some sort of systemic disease. And so my level of concern was growing because, again, I was hoping to use these fish for experimental challenges. So I needed to know what was causing these infections. And so I began to employ standard diagnostic protocols in order to diagnose the problem. So following virological analyses, I wasn't able to detect any virus infection. At the same time, I started looking for bacterial infections. So I took cultures from the kidney, but because these fish were showing signs of severe exophthalmia, I also took bacterial cultures of the eye and the brain. And in many instances, I was getting this pure, intense bacterial infections. And again, these were all recovered from the kidney, the eye, and the brain of these fish. And so I used the same approach as adults and performed bacterial analysis and reached a presumptive identification of this bacterium belonging to the genus Carnobacterium. And then again, to confirm its identity, I ran the same PCR analysis as I did before, analyzing a range of representative isolates. And once again, sequence analysis showed the bacterium belonged to genus Carnobacterium. And then I performed phylogenetic analysis, adding all of my isolates that I collected from the disease outbreak in the lab to the dendrogram I showed you earlier. So once again, all of my isolates fell into this robustly supported clade that belongs to the type strain of Carnobacterium multormaticum. So unfortunately, antibiotic treatment was not an option because remember, these fish were destined for experiments with bacteria. So in the meantime, I continued to pay utmost attention to rearing hygiene, and I employed non-chemical treatments like salt baths. Ultimately, when the mortality stopped, I was left with a cumulative mortality ranging from 25 to just about 32% in each tank, with a median of 28% cumulative mortality, meaning that this disease outbreak resulted in a substantial mortality. And at this point, I knew I couldn't use these fish for challenges, so I sampled fish from each tank to understand how prevalent this bacterium was. And again, these mortalities seemed to stop. And while examining these fish, and although very rare, I was still finding some severe signs of disease, including ascites taken from fish. And so about 1 week after the mortality subsided, I randomly sampled 60 fish from each tank. And so the prevalence was relatively low, so ranging from about 3 to 6%, providing further evidence that the disease outbreak had subsided. However, I was still able to detect Carnobacterium infections in the kidney, the eye, and most importantly, the reproductive tissues of these juvenile fish. And then I started having discussions with Dr. Isaac Standish, who recently developed a qPCR assay specifically for this bacterium. So I sent him tissues of an additional 60 fish from each tank. And again, although using a different method, and now this is about 2.5 weeks later, we're still seeing the prevalence is approximately between 3 and 6%. So once again, the outbreak had subsided, but we're still finding these infections present and in multiple tissues of infected fish. And of note, I also found the bacterium within the swim bladder fluid in the lumen of 2 fish. And if you can recall from earlier, I found the same growth sign in wild adults. So yeah, 2020 came with a ton of challenges, one right after the other, but when life hands you lemons, and in this case, 2020 hands you lemons, I had to learn to make lemonade. Because even though I couldn't use these fish in experimental challenges, I was still able to understand what was causing this disease outbreak. And I discovered that it was one of my target pathogens, and so I was able to collect data. And after all, the result of this was the first Carnobacterium disease outbreak in Great Lakes lake whitefish under laboratory conditions. So some overall takeaways is that I found Carnobacterium in diseased adults. And I also found Carnobacterium in the reproductive tissues of wild spawning adults. So again, some showing signs of systemic disease. So this creates that potential pathway from the pathogen to get from the infected parents to its offspring. And what's notable is it's notable, especially when I'm talking about fish in early life stages doing so poorly. I also found in 2020 that the bacterium led to natural infection, which shows that Carnobacterium can clearly have negative health effects on juveniles. And so this is something that we didn't know to this point. So now to come full circle, we see this is a very complex situation involving environmental factors that are suboptimal, like those that we're seeing in Lakes Michigan and Lake Huron, causing the host, Coregonus clupeiformis, to become stressed. And this is either related to those environmental conditions or food web changes or other factors. So this opens the opportunity for which could be causing them to become susceptible to infectious pathogens, like Carnobacterium multormaticum. And so when these factors interact like we see them interacting now, this creates the perfect opportunity for disease to occur. So I'm not saying that this is a primary cause driving the declines in recruitment. However, this could be one factor. thus creating the potential for the infectious disease to have a negative effect on the health of lake whitefish, and subsequently negative effects on the recruitment of lake whitefish to the Great Lakes. So at this time, I'd like to thank the Great Lakes Fishery Trust for funding this project, the fishing boat captains for helping me collect the adult fish, MDNR Fisheries Division and Fish and Wildlife Service for helping me collect the juvenile fish, the Little Traverse Bay Bands of Odawa Indians for supplying the fish used in experimental challenges, my friends help along the way, Dr. Isaac Standish, all of my colleagues at the Michigan State University Aquatic Animal Health Lab, and my advisor, Dr. Thomas Locke.
All right, thank you, Courtney. A very interesting project and excellent presentation.
Thank you.
Feel free to unmute yourself and ask Courtney a question, or you can put it in the chat box. Esteban, I see your hand. Go for it.
Hi, Courtney. Great talk, and thank you for presenting so eloquently. I wanted to ask you, I think I asked you this before, and you mentioned environmental conditions, that could have been potentially stressful for these fish. For a lot of the, and I know this is not a streptococci, but behaves, very similar to other streptococci like lactococcus and streptococcus ini and streptococcus agalactiae. A lot of the times, we see them with higher prevalence, particularly in salmonids, as they get temperature stressed. So we're getting our behinds kicked, with Lactococcus garvey in California, particularly when the salmonids reach, above 18 degrees Celsius in temperature. So you mentioned the environmental conditions, and I think I had asked you this, between the 4 different recruitment sites that you investigated, do you have evidence, or did you measure any water quality or at least temperature comparisons, to see if for every reason, like the sampling site would form, some sort of a bay or something like that where water does not circulate and can increase? I'm just curious about that. Did any temperature was measured in any of those sites?
I mean, I didn't take the temperature whenever I collected these fish, but that is, something I could easily look back at, like, water temperature. I could easily find that information, but yeah, I mean, another aspect of this study too is once I'm finished with pathogen studies is to take juveniles and again infect them with these pathogens that I'm finding in the adults and see what happens at different water temperatures. Also with different host densities too, because I know that could also be a factor in causing stress to these fish.
Yeah, no, absolutely. And I'm just curious. I mean, I know that you have been finding these organisms, particularly in whitefish, that is what you're looking at, but perhaps, for the model of infection, have you tried in other salmonids, like in rainbow trout or in salmon? Because sometimes, this bacterium, the specificity might not be as big, perhaps, if you have trouble getting whitefish, you can try it in other salmonids.
Right, yeah, and I was actually, talking to my advisor, Dr. Locke, about this yesterday, how, Corynebacterium is found in many fish populations, so it would be really interesting to gather some Corynebacterium isolates recovered from these other salmonid species and compare them to what we're finding in Great Lakes like whitefish.
And as far as typing goes, I mean, I know that you did sequence of the 16S, but are you guys planning on doing a little bit more genotyping for a little bit more, maybe more housekeeping genes, or at least other profiles? Rep PCR could be another good one, just to get a sense of are these really all the same strain or different strains of the species?
Right. Yeah, and thanks for bringing that up. So we actually haven't had any plans on genotyping these isolates just yet, but obviously now there is discussion about it. So I could see that, after this project, that could continue on to future work with Corynebacterium and salmonids in the Great Lakes and and other places too. Yeah, so
Excellent.
thanks for bringing that up.
Great job, Courtney.
Thank you.
Great job.
Hi, Jim, I see your hand up too.
Okay, thank you. Courtney, that was really interesting. It's somewhat reminiscent of some of the R. salmoninarum or BKD work in Pacific salmonids, and it really, I thought your fish that you were trying to rear for pathogen-free experiments are telling you almost certainly that there's vertical transmission of this bacterium. And while it's not necessarily the definitive experiment, you may have opened up a really interesting piece of work. Is it possible to hold those fish to adulthood and to get them to spawn? And in case you could then look at interovum presence of the bacterium, or by sampling individual fish, you might be able to develop one or two of the fish that are in fact pathogen-free, as they did with our salmoninarum, where they screen the adults and hold eggs from the negative adults to develop your pathogen-free stocks.
You know, thank you, Jim, for asking that. And I would love to do something like that. Unfortunately, from what I've learned from rearing these lake whitefish over the past 2.5 years, is that they are very finicky and they're hard to rear. So, I mean, I think it's possible to grow these to adult size and then keep them and have them for research purposes. But from my own experience, you can just look at one of these fish and then the wrong way and they die. So it's very hard to rear these in like a hatchery setting. But that is a great idea.
Well, then it may be possible to look at natural fish with a qPCR or other assay to try to identify adults that are negative that you could use for your next experiments.
Yeah. Yeah, thank you.
All right, any other questions out there?
If there's not, I'm just gonna follow up, Jim. Courtney, you'll recall that we had a similar conversation, right? I can come up with all sorts of crazy ideas and piling stuff. Jim, your idea is not crazy, but I'm saying I was absolutely trying to coax Courtney into, what do you think about, could we try and salvage some of these fish and try and maintain our own captive brood stock? So we didn't take that path, but I'm with you. I think as we learn, it might be the only way where we're going to be able to get pathogen-free fish, other than maybe qPCR, like you're saying. But I still am worried, even that path.
Well, Tom, you don't need to apologize. Most of my ideas are crazy. You can ask any of my students or in the past or my colleagues. Often wrong, but never in doubt, as they say. So.
Good stuff. All right, excellent job, Courtney. Virtual round of applause to you.